Okay, guys, welcome back. Today we're going to be talking about the lymphatic and the immune system. They're always mentioned together, the lymphatic and the immune system, because they cooperate anatomically and functionally to help us achieve homeostasis. We mentioned one thing about the immune system, and that is the immune system is a collection of different structures like your antibodies, like your innate immunity, like your adaptive immunity, and they work together to ensure you have protection against microbial infection. And we're also going to learn that this immune system can sometimes go wrong and it will attack your own body. And that is known as autoimmune diseases. That is when your immune system has amnesia. It no longer is able to recognize the difference between self and non-self. So the lymphatic system is a very important system that cooperates both with your cardiovascular system and your immune system. So it's in the middle between the cardiovascular system and the immune system. And we talked in lab about how this cardiovascular system cooperate with your lymphatic system. Does somebody remember? Does somebody remember the significance of remember when I drew that we have two circulations? We drew that we have two circulations, one red, one red circulation, and one blue circulation. Does somebody remember? Does somebody remember the red versus blue circulation? This is very important. We said that we have red blood coming in. Somebody have a red marker? Red blood coming inwardly, and we have red blood coming in, and we have what color blood coming out? We have blue blood coming out. And we mentioned something very special. Okay. We could have, okay. So for example, let's assume that this, I do not have red color. Let's assume that is brown here is the red part of the circulation. You see? This is red blood that is rich in oxygen, rich in nutrients, and you have blue blood coming out the other direction. You have blue blood coming out of the other direction. What did we name these holes here so what is this whole microcirculation called? This is known as micro, this is the micro circulation. This is the microcirculation. And we said that in the bottom, what do we have down here? What do we have down here? What do we have down here? What do we have available? What do we have exuberant and available down here? So these are tissue. These are tissue compartment, and that is our microcirculation. So when the blood arrives to our tissue, what happens? Oxygen, oxygen gets out. We have oxygen coming out. We have oxygen coming out, and we have nutrients. We have nutrients. We have ATP coming out to the tissue. What is the name of the force that powers the movement, this dialysis? Remember when we did the dialysis bag experiment in lab? That's exactly what's happening at your tissue compartment. This filtration, filtration, this filtration, this dialysis, this dialysis, this filtration, this dialysis obviously has to be powered by energy. We cannot have molecules moving from one place to another without energy. What is the energy that powers the filtration movement, the dialysis? What energy is that? That is kinetic energy in the form of hydrostatic pressure. Hydrostatic pressure. This hydrostatic pressure, who powers it? Who causes the hydrostatic pressure? Who's responsible for generating? Who's the generator for hydrostatic pressure? The heart. So your heart generates hydrostatic pressure. And out through these fenestration pores, the oxygen comes out to nourish the tissue. Then we have the return. We have the return of all of the negative solutes, negative carbon dioxide, okay, the negative metabolic waste, the negative nitrogenous waste. All of those are leaking back into the holes. They are leaking back into the circulation. What is the movement caused by? This leakage, so we said that to come out from the red part of the circulation into the tissue, that is powered with the heart, the hydrostatic pressure. What pressure? This is filtration pressure on the red side. On the blue side, what kind of pressure is that? What kind of pressure is help bringing back the solutes, bringing back carbon dioxide from the tissue into the microcirculation? What's the name of that particular type of pressure? That's the osmotic, or also known as, very good. That is the osmotic, also known as, osmotic pressure is also known as colloid. Osmotic, colloid, or oncotic pressure. And that brings back all of the solutes and the waste and the carbon dioxide from the tissue compartment into the capillaries. Now, we said that the exit of all of the beneficial oxygen and nutrients to the tissue is 100% through the filtration. But when we have all of these negative toxic waste, only about 85% returns. There is some minus 15% that lags behind. There's a buildup of 15% carbon dioxide, 15% negative solutes, 15% metabolic waste, 15% nitrogenous waste. Who's going to restore that back to the circulation? We're going to leave it behind like that? We're going to leave it behind building up in the tissue? So how are we going to restore it back? We said that in the lab. How are we going to restore back all of this minus 15% of metabolic waste tissue that is lagging behind? Is your lymphatic system. So your lymphatic vessels are going to take all of this 15% that did not make it into the microcirculation through the oncotic, osmotic, colloid pressure that's going to build up in the back. Your lymphatic system will happily take it back. So this is a safety line. This is a safety line. Your lymphatic system is a safety line that rescues all of the tissue compartment waste that did not make it through this weaker. When you compare filtration, hydrostatic pressure is very effective. It gets out 100%, but the oncotic colloid osmotic pressure only takes 85 and leaves behind minus 15. That minus 15 is restored and taken back with your lymphatic system. Okay, so now let's look at the anatomy of our lymphatic system. They always refer to it in the green color. If you examine here, you would see that the lymphatic vessels are found in the arms, the legs, and they convey lymph fluid to the larger lymphatic vessels in the torso. So this lymph fluid is made up of what? It's made up of a mixture of all the fluid that is lagging behind in the tissue compartment and unable to return through the osmotic, the colloid, the oncotic pressure back into the microcirculation. Now, if you look at these green vessels, if you look at these green vessels, what do you see here in these if you population, what are these dotted circles representing? This is your tissue here. So you see you always have red vessels. They should also have what color vessels here? Blue. You see, you have red blood vessels bringing in oxygen, nutrients to the tissue compartment, and you also have the blue vessel. They did not draw them, but they just refer to them as these red vessels. But there's blue vessels that are taking back. Guys, this is really important. It's very important for you to know that the microcirculation is made up of two kinds of vessels. Red vessels, I drew them here in brown, but they're actually red. They get out oxygen, they get out oxygen, and they get out ATP, they get out oxygen, and they get out ATP and nutrients. And when they use the oxygen for oxidative, why do we need oxygen? We need oxygen for oxidative phosphorylation. We need oxygen for aerobic respiration. And then we're going to have some waste that is building up. You don't just order Uber Eats and you order pizza. You need to, after you eat that pizza, you're going to throw it in the garbage. So you now have to have a system, a sanitation system to pick up all the waste and the garbage back. So what is the pressure that is guiding the movement of the supply to your tissue here? Good stuff is coming out from the microcirculation to the tissue through what kind of pressure? Hydrostatic or filtration pressure. And the removal of the waste back into the microcirculation is driven in part by what? By the osmotic, colloid, oncotic pressure. The same thing, tomato, tomato. Colloid, osmotic is the same thing. Colloid, osmotic, oncotic pressure. These are all different types of pressure. These are all different types of, they all different words meaning the same thing. But this colloid osmotic pressure is like magnet, sucking and removing all the carbon dioxide metabolic waste from the tissue. But it's only removing 85%. There is 15% that's failing and lagging behind. Who's carrying that backward? That is your, who's carrying that back? That is your lymphatic system. So the lymphatic system here, you see this green line here? This is all, you see this node? The node is the headquarter. So we pick up all of the nasty stuff from the microcirculation and we transfer it back to the node. So we have a lot of lymphatic vessels and we have a lot of lymphatic nodes. So for example, that's a type of a lymph node. The spleen is a huge lymph node, and you have the thymus. The thymus is the lymph node in which the T cells grow. So why do we have B cells? B cells, they mature in the bone marrow, while T cells mature in the thymus. So this thymus here is a lymph node that is going to be the final site for the maturation of the T cells. That's why they call them T cells. While the B cells come out already mature from your bone marrow. So that's exactly what we drew here. So if you look here, you see that the red blood is coming in. You see in the back, you see these dots, these dots. That's why I drew here. These dots here. These dots are your tissue compartment. These dots are your tissue compartment. Who is bringing them red blood? It is your red side of the microcirculation, your arterial side. And the blue side is removing through colloid oncotic pressure all of the minus stuff and bringing it back into the venous circulation. The green line is helping. Who's this green vasculature network helping? Are they helping the red side or are they helping the blue side? Yeah, they're helping the blue side. Why are they not helping the red side, but they're only helping the blue side of the capillary microcirculation? Yeah, the remainder of the carbon dioxide and the nitrogenous waste. Now, this is showing you that the fluid that flows inside the lymph vessels is known as lymph. The lymph fluid, all the stuff that lags behind, is passing inside these tunnels known as the lymphatic vessels. The same way you have endothelial cells that make up the blood vessels, you also have lymphatic vessels. These are lymphatic vessels. And these are endothelial flaps. These flaps open up to allow for the entry of the lymph fluid. What are these dotted lines representing? These dotted circles. They represent what? These here. You see, I drew them here. They have dots. What are these? That's the tissue. So you see, any waste that lags behind in the tissue is going to squeeze and enter through these. Is going to squeeze and enter through these flaps. Through these flaps. Okay. This interstitial fluid. What is the interstitial fluid and what is the intracellular fluid and what is the extracellular fluid? So we know inside the cell, how is water divided in our body? When we have cells, the cells represent tissues. So multiple cells make tissues. Multiple cells make tissue. So the cell is bathed, is bathed in water. Is bathed in water. So the water inside the cell is known as the ICF or intercellular fluid. And between the cells, you have intracellular fluid. So the water inside the cell, that is intracellular fluid. Then outside, you have extracellular fluid, ECF. This extracellular fluid is divided into interstitial fluid and intravascular fluid. Intravascular fluid is the fluid inside your blood vessels, inside your vasculature. Inside the vasculature, you have the intravascular fluid. And now we're not talking about inside the cell no more. We're talking about between the cells and the blood vessels. That is the interstitial fluid. So this water here, this water that is not inside the cell, but rather bathing the cell from outside, and it is not inside the blood vessel. So water can only be either inside the cell or inside the blood vessel or bathing the cell from outside. So what's the name of the water that is inside the cell called? Intercellular fluid. The water that is between the cells and bathing the cell from outside but not inside the blood vessels. What is that known? Interstitial fluid. And the water that is inside the blood vessels is known as the intravascular fluid. And when you combine both interstitial fluid and intravascular fluid, both, what is that called? Extracellular fluid. Excellent. So you have your endothelial flaps, and inside the inside the lymph vessel, the same way we talked about how inside the veins, inside the vein, you have, when we talk about varicose veins, when we talk about varicose veins, when we talk about varicose veins, in varicose veins, you're supposed to have valves. These valves actually, they look like this. The valves look like that. Why do they look like that? Because your foot is under here. So the blood flows up through the valve, up through the valves. If the blood is trying to fall back, if the blood is trying to fall backward, these flaps, these vessels are going to prevent it, are going to fight back to prevent the blood from falling inward. So they're gonna reverse the blood and make sure they're only one-way valves. That's why when these valves are defective, when these valves are destroyed, they become very weak. So now they allow reflux. They allow reflux of blood, so the blood is gonna fall back and the blood is gonna pool. You're gonna have pooling of blood. You're gonna have engorgement. You're gonna have engorgement of blood vessels. You're gonna have engorgement of blood vessels. Why? Because the valves have been defective. They're supposed to be one way, allowing the exit in one way. But they have been, due to obesity, due to pregnancy, they have been damaged, and now they allow the blood to flow back in the opposite direction. Now, when you look at the lymphatic vessels, so now this is no longer a blue vein. What color did we say the lymphatic vessels are? The lymphatic vessels have green color. They have flaps. They have flaps. They have flaps. What do the flaps do? These are lymphatic flaps in both directions. Lymphatic flaps. These are known as lymphatic flaps. What do the lymphatic flaps do? They remove, this is our tissue with so many cells, they remove all the lagging behind, all the solutes, all the nitrogenous waste, okay, all the metabolic waste known as lymph, is known as lymph. They allow all the lymph to enter through the flaps. Now, once the lymph enters through the flaps, there's one-way valves. The one-way valves allow the passage. They allow the passage of lymph in one direction to go back into the veins. So the lymph is going to be deposited in the veins so it can enter into the right atria. It can enter into the right atria. Then from the right atria, we can enter into the RV, the right ventricle. And then we get pumped into the lungs, and the lungs is gonna oxygenate all the deoxygenated blood and refresh it and make the actual gas exchange, and then we're gonna have red blood coming back. So the lymphatic fluid also always fall into the subclavian vein, and that is a blue vein to go back in because that's waste. The vein is carrying the oxygenated blood, and we said the green lymph is helping the blue. So they always pass together, and then they dump all of their content into the subclavian vein. Then from the subclavian vein, we fall into the right atrium, into the vena cava. From the subclavian vein, then we have the vena cava, and from the vena cava, we go into the right atrium. So these are the flaps, and these are our valves that prevent the backflow. That's our interstitial fluid, and we need the collagen fiber to give structural tensile integrity for the cell. So we have the collagen. So this collagen it's a protein. We said collagen is what kind of protein? Primary, secondary, tertiary, quaternary. What kind of protein is collagen? Yes. Collagen, and who else is also secondary besides collagen? Keratin. Excellent. So collagen and keratin are both secondary structure proteins, and they give tensile integrity for the structure and architecture of the cell. So that is part of our tissue compartment, collagen. It gives it this elasticity, this malleability to be able to bend. Your tissue is able to bend without breaking. This extensibility, the malleability of your tissue. So the lymphatic capillaries are interlaced. You see, they are interlaced with the arterioles and the venules of the cardiovascular system. And the collagen fibers anchor a lymphatic capillary in the tissue, and the interstitial fluid slips through the spaces between the overlapping endothelial cells that compose the lymphatic capillary. What is the job of these flaps? Can somebody please tell me what is the job of these flaps in complete sentence? Yes.
Protect out all the trash of the body.
Yes, so it's good what you're saying, but when we say trash of the body, take your time. Yeah, to remove all the waste and the carbon dioxide. The waste is you have nitrogenous waste, metabolic waste, they're going to enter through the flap. Now, this internal valve here, what is the function of it and what is the structure of it? This valve. I want two answers. What is the structure and what is the function of this valve? The structure is a one-way or a two-way valve. One-way valve. You know, sometimes they have a door that open one way. Sometimes you have a door that open both ways, right? So that's a difference in structure. One structure, one way, one structure, two ways. So the structure here is a one-way valve. Now, what is the function of this valve that is one way inside here? Yes. Excellent. That's a complete answer because we want all the fluid to move in one direction into our veins, into our subclavian vein. Then after the subclavian vein, where are we gonna go? After the subclavian vein, where are we going? Superior vena cava or inferior, because you know we have two types of body, upper body, lower body, but you got it. The superior vena cava and inferior vena cava. Then after the superior vena cava, inferior vena cava, where do we go? We go to the right atrium. first. Then after the right atria, where we go? Right ventricle. Excellent. Then after the right ventricle, where we go? Pulmonary circulation. Then we go back into the left heart, left atria, left ventricle, aorta. Then it's excellent, excellent, excellent. Good job. Okay, now these are the major trunks and ducts of the lymphatic system. So there's a thoracic duct that drains a much larger portion of the body than does the right lymphatic duct. How many ducts do we have, guys? We have two ducts. There are only two ducts. This purple duct is known as the right lymphatic duct. Look at what it drains. It drains your right arm, it drains your shoulder, your axilla, and the right side of just this purple part. It is known as the right lymphatic duct. And it dumps all of its lymph inside the right internal jugular vein. And then the right internal jugular vein is obviously going to mix with this right subclavian vein, and both are going to be into your superior vena cava. You see, that's your superior vena cava. So part of the lymph is going to be deposited into this here. What is that? That's your right subclavian vein. And more lymph is going to be deposited into the right jugular vein. And all of the lymph from the right subclavian vein and the right jugular vein, both are going to be converging to enter into this big blue vessel entering the heart. This is known as yes, what is that? Superior vena cava. Excellent. Now, this is only for the right duct. That is your right thoracic duct. This is your right lymphatic duct. The blue is right lymphatic. Now, this whole rest of the body in yellow, that is known as your thoracic duct. That is your thoracic duct. So the thoracic duct empties into the left subclavian vein and the left jugular vein. And both of them, they're going to drain into the subclavian. And then you're going to have this branch that's going to take all of the lymph that is taken from the thoracic duct into what is the name of this big vessel? This is known as the same one as this one. That is your superior vena cava. And then you're going to have all the blood following through. So the thoracic duct. So you need to know that the right duct, this right lymphatic duct, drains a much smaller region of our body compared to the thoracic duct that drains the rest of our body. So yellow is thoracic duct and purple is what duct? Right lymphatic duct. Okay, so now when we look at our bone marrow, all the cells of the immune response, as well as of the blood, arise by differentiation from hematopoietic stem cells. Platelets are cell fragments involved in the clotting of the blood. We talked about that. So you have to understand the concept of the cytokine. What is a cytokine? A cytokine is, when we want to define something, what is it? What are we trying to define? We begin very big. Is it a cell? Is it a tissue? Is it an organ? Is it an organ system? If it is not one of those, then we move smaller. Then it's obviously a subcellular compartment. Subcellular compartment can either be an organelle, it can either be a macromolecule. So what do you think a cytokine is? So a cytokine, it is not an organ, it is not a tissue, it is not an organ system, it is not an organism, it is it a cell organelle? Is the cytokine, when we studied cell organelles, We studied the plasma membrane. We studied the Golgi body. We studied the nucleus. We studied the ribosome. Did we study an organelle called cytokine? So it is not an organelle. So what is left? What else could it be? What else did we study? What else did we study? What did we study? We studied organelles. None of them was called a cytokine. So it's not an organelle. We studied cells. Did we hear of a cell called a cytokine? So that is not a cell. We studied tissues, the different kinds of tissues. Remember? We studied the epithelial tissue, the connective tissue, the nervous tissue, and the muscular tissue. Did we study cytokine tissue? So it's not a tissue. We studied organs. We studied the liver, we studied the muscles, and we studied the brain. And it is not an organ. We studied organ system. It's not an organ system. It is not an organism. So what is below organelle? Yes. Enzyme. Excellent. So now we're heading in the right direction. But enzyme itself, what is it? What? Is a protein. Excellent. So a cytokine is a protein. So always think when we move down from organelles, enzyme is a type of proteins. So below organelles, you have macromolecules. Macromolecules. These macromolecules are known as lipids, carbohydrates, and proteins. What's the last family known as? What's the last family known as? Nucleic acids. Okay, so proteins. These proteins, they are very important. Cytokines. What is their job? Well, one type of protein was an enzyme. One type of protein was an enzyme. What does the enzyme do, Julie?
It speeds up chemical reactions.
It speeds up chemical reactions by decreasing the energy barrier. Does the cytokine do that? It does not do that. It does not act. It's not a catalyst. This is called catalysis. Enzymes are there for catalysis. Cytokines don't do no catalysis. So what else can they do? They are.
They act as the immune system, like depending.
So yeah, they are messengers. But what kind of messenger? They are ligand. That's the word I was looking for. Remember when we said acetylcholine? Acetylcholine would bind into the channel, causing the channel to open up. Causing the channel to open up to allow for influx or efflux. That's the ligand. That's the key. You put it in, it's gonna open. But not every ligand is gonna open a channel. We heard some ligands. Remember when we looked at the cell membrane, we said that some ligands, when they bind, they do what? Not all the ligands, when they bind, they open a channel. What else can ligands do? They can cause a conformational change that's gonna, like, remember the G protein? It can go and it's gonna elicit a second messenger, and then it can go phosphorylate an enzyme to give you a target response. That's another ligand. But cytokines, they do just that. They bind to a receptor. So you have a primordial cell. You have a multi-potent cell. A multi-potent cell, that is a multi-potential. It has multiple potential. When you were a kid, you had the potential of becoming a police officer. You had the potential of becoming an athlete in the NBA or the NFL or whatever. You had a potential for becoming a firefighter. You had a potential for becoming a doctor. So you have multiple potential. Same thing. The cell here, it has the potential to become a platelet. It has the potential to become red blood cell, erythrocyte. It has the potential to become basophil. It has the potential to become neutrophil. It has the potential to become eosinophil. It has the potential to become monocyte. It has the potential to become natural killer cell. It has the potential to become T lymphocyte. It has the potential to become plasma cell. Now, who's going to make that potential happen? The cytokine. So there's a cytokine that once it is bind to a cell, you give it that cytokine, it becomes this platelet. You give it platelet cytokine, it will become platelet. You give it erythrocyte cytokine, it will become erythrocyte. You give it basophil cytokine, it will become basophil. You give it the neutrophil cytokine, it will become a neutrophil. You give it eosinophil cytokine, it will become eosinophil. You give it macrophage cytokine, it will become macrophage. You give it natural killer cytokine, it will become natural killer cell. You give it the cytokine for small lymphocyte, it will give you either T lymphocyte or B lymphocyte. The cytokine is the transcript. You know, when you give the pharmacist the transcript, he will give you antibiotics, he will give you pain medication, he will give you all the different kinds of medication based on the transcript. So the cytokine is a protein that is acting like the protein that acts like a transcript. You really need to know that. And some cytokines, if we look here, some cytokines, these cytokines, for example, of bacteria, invade your body. A virus invades your body. If a parasite invades your body, if a worm invades your body, if they all invade your body, on the surface, you know, in the crime scene investigation, remember CSI? In the crime scene investigation, when a crime happened, what the police look for? The fingerprint. They look for the hair, your DNA, because these are things that trace back to you. So the same thing, the bacteria doesn't have fingerprint, it has protein on the top, it has sugar on the top, it has lipids on the top. So that is gonna fall off inside your body. When it falls off inside your body, it becomes a cytokine. So you have your immune cells, these immune cells, your T cells, your B cells, they have a receptor that will catch these things that are cytokine that falling off. These cytokines that are falling off from the bacteria. So they have cytokines falling off from the bacteria. So if you look at these cytokines, they can be secreted by your own body for multipotential cells, or they could be used for alarm signal, different kinds of cytokine. Some cytokines, they trigger inflammation. Some cytokines, they trigger inflammation. Some cytokines, they trigger the catching of these different types of these different types of infectious microbes. So here, our multipotent hematopoietic stem cell, known as the hemocytoblast, has one of two potentials. The first potential, it can give me more reserve. It can give me more reserve of multipotent hematopoietic stem cells, and it could give us remaining cells that can wait for the chemical. You see here, what does it say? The remaining cell goes down one of the two paths depending on the chemical signal received. What's the name of this chemical signal? Excellent. Excellent. So the cytokine, when it touch, there is one cytokine, when it touch this cell, it becomes lymphoid stem cell. There is one cytokine. When it touches this cell, it becomes myeloid stem cell. So this myeloid stem cell, again, one cytokine can make it become a monoblast. And what do the monoblasts give us? Give us a monocyte. And the monocyte give us macrophages. The monocytes give us macrophages. There are also myeloblasts. These myeloblasts, there are cytokines that give us basophils. There are cytokines that give us neutrophils. There are cytokines that give us eosinophils. There are also cytokines that give us reticulocytes, and the reticulocytes, these are the precursor cells for your erythrocytes. So you guys need to know this. Who's the precursor cell for the macrophage? The monocyte. You guys remember the Pokemon? You have the little Pikachu that's gonna grow up and become into. That's exactly what happens here. But in the Pokemon, it doesn't need a cytokine. It just happens with time. But here, you need a cytokine. So your monocytes, they are circulating inactive. But once they detect, you know, they like a cytokine. When they detect a cytokine or when they detect a bacteria, they differentiate into a macrophage. You see, they different into a macrophage. What are the precursor cells for the basophils, the neutrophils, and the eosinophils? These are your myeloblasts. What are the precursor cells for the erythrocytes? The reticulocytes. The reticulocytes. So on the exam, I'll ask you the reticulocytes. Give us what kinds of cells? Basophils, erythrocytes, megakaryocytes, monocytes. Erythrocytes. What are the cells that give us platelets? Megakaryocytes. So this entire myeloid lineage will give us these defense cells. But the lymphoid lineage will give us your T lymphocyte and B lymphocyte. So these are your specific immunity, your T lymphocyte. But there is something very interesting in this lineage, and that is you see these NKCs, natural killer cells. These natural killer cells, they come from where? Do they come from myeloid lineage or lymphoid lineage? They come from lymphoid lineage, but their job is with everybody else. So all of these guys, they read the basophil, the neutrophil, the eosinophil, the macrophage, they are important in generalized immunity. And they come from myeloid. But the natural killer cell, they come from lymphoid and they still do generalized immunity. Even though they come from specific immunity lineage. Okay, so what's the difference between general immunity and specific immunity? You know the police, you call 911, the police will come for robbery, they come for assault, they come for a fight in the street, they come for disorderly conduct, they come from general things. But when the problem gets bigger, then you have the detective. The detective will come for fraud, for homicide, will come from these. So these are the detectives. The T lymphocyte and the B lymphocyte, they have specific function. These are detectives. They come later. That's called specific immunity. But these guys here, the basophil, the neutrophil, the eosinophil, and the natural killer cell, this one is with them, even though it comes with the question they ask on the exam. What is so special about the natural killer cell? What's so special is that they do generalized immunity just like the basophil, the neutrophil, the eosinophil, the macrophage, but they do not come from their mom, the myeloid stem cell. They come from the lymphoid lineage. So they come from the same mom, they have different jobs. These guys become specialized, and this one becomes just like these guys with a step. These are step siblings. So this is generalized, and this here specific. Very important to know that. We have generalized immunity to fight all general infectious microbes. Fungi, bacteria, removal of necrotic debris, damaged tissue, parasite. We remove things generally. But when things get really severe or virulent diseases, you have your lymphocytes that will come, the T lymphocytes and the B lymphocytes. Okay, the bone marrow. Why? Remember when we said, when we study histologically, you cannot study from one angle. You have to slice the egg in half. If you just study the egg by looking from the outside, you will never know there is yolk inside. You will never know it's yellow inside unless you cut it in half. You know your friend. You know somebody. You meet somebody. You only know one side of them. Like the actor. They have personality for acting in front of the people in the auditorium, and they have a backstage. They have a front stage personality and the backstage. If you're not with them in both, you will not know their true personality. So the same thing, this bone, if you don't cut it sagittally, you will never know what is inside. So when they look inside, they saw not only white osteoid tissue, periosteal tissue from the outside, there's actually red and yellow. Why do we have red bone marrow? Red blood cell. Why do we have yellow marrow? Lipids. Why we need lipids? Energy. Very good. Good job. Today I accomplished my goal. Okay. We talked about the thymus. This thymus undergoes something known as involution. Involution. What is involution? What is involution? This is involution. Involution means what? The thymus look like that when you are born, when you are 15 years old. The thymus looks like that. When you are 80 years old, your thymus looks like this because it underwent involution. So what does involution mean? Shrink, to get smaller. And we said that the thymus, that this thymus is the site for the maturation, the site for the maturation. Maturation of what? Excellent. So we call them the T cells because they mature in the thymus. So the thymus, as you get older, become 80-year-old, they get smaller. When they get smaller, what happened to your immunity? When your thymus gets smaller, what happened to your immunity? That's why the elderly people, they have a weak or a strong immune system. They have a weak immune system. Okay. So the thymus, when you wear a necklace, where the necklace exactly falls, that is where the thymus is. So the trabecula and the lobules, including the darkly stained cortex and the lighter staining medulla of each lobule, these are clearly visible in the light micrograph of the thymus of a newborn. Yeah. So do you see that this is your thymus? It looks like that. See the structure? That's the thymus. So it's yellowish in color and it has these lobules. It's yellowish and it has thymus is right above your heart. And you see you have right lymphatic duct, and these are the adenoids and the tonsils. These are lymph nodes. The adenoids and the tonsils, these are lymph nodes. And you have this cortical epithelial cell, and this is you have in the thymus, you have a trabecula, and within the trabecula, you have your thymocytes. Thymocytes are immature what? Thymocytes are immature T cells. And you have these are the blood vessels bringing in blood, oxygen, and ATP. And you have dendritic cells. These dendritic cells, these are cells that are, this is the medulla, that is the cortex, and that is the fibrous. You see, that's the fibrous capsule. That's the cortex, and that's the medulla. You see, that's the structure and the histology of a lymph node. So that shows you your lymph node. There is an efferent lymphatic vessel, and there are afferent lymphatic vessels. So the efferent, they bring in your lymph fluid into the node. So you have to make the connection and the difference between efferent and afferent. Efferent and afferent lymph nodes. There are efferent and afferent lymph nodes. There are efferent and there are afferent lymph nodes. So you see, if you are entering into the lymph node, coming in, efferent. Why are they called efferent lymph vessels? Because they're going to the effector. Effector. The effector, who's the effector? You know, when you call 911, you want the police. The effector is the police officer. You call 911, you want a fire truck. The effector is the fire truck. If you want the ambulance, the effector is the ambulance. The effector is the one who does the effect that you're seeking. So here the effector is what I'm trying to get. I'm trying to get into my lymph node. This is my lymph node where I have all the filtration happening inside the lymph node. So if I'm coming in, that's efferent vessels. After I get filtered out, I'm gonna have afferent vessels. These are, you see the afferent lymphatic vessel. So lymph nodes are masses of lymphatic tissue located along the larger lymph vessels, and the micrograph of the lymph nodes shows a germinal center which consists of rapidly dividing B cells surrounded by a layer of T cells and other accessory cells. Okay, so now we're going to look into the spleen, which is a major node. But don't forget that efferent lymphatic vessels bring in the lymph inward into the lymph node, where you have trabecula and you have the cortex. The cortex is the top part. Even in the brain, you have the cerebral cortex. In the kidneys, you have your nephron cortex. In the lymphatic vessels, you have the lymphatic cortex. Cortex is the surface of the organ. So lymph nodes are masses of lymphatic tissue, filtering tissue along the larger lymph vessels. The micrograph of the lymph nodes shows a germinal center which consists of rapidly dividing B cells surrounded by a layer of T cells and other accessory cells. The spleen is attached to the stomach. So a micrograph of spleen tissue shows the germinal center. The marginal zone is the region between the red pulp and the white pulp. So the red pulp is where we make the red blood cells. And the white, you see, that's the red pulp, and that is the white pulp, which sequesters particulate antigens from the circulation and presents these antigens to the lymphocytes in the white pulp. So now let's look at the tonsils. We have two types of tonsils. There are pharyngeal tonsils. The pharyngeal tonsils are located on the roof of the posterior superior wall of the nasopharynx. How many different pharynx do we have in terms of order from the top to the bottom? You have your, yeah, which are the what pharynx? Nasopharynx. Then you have the oropharynx. Then lastly you have the laryngopharynx. Very good. You have your laryngopharynx. And then you have palatine tonsils. So the tonsils, they are located here. You see the palatine tonsils? And you also have lingual tonsils that are found here. These are the lingual tonsils and the palatine tonsil. These are the palatine tonsils. And right behind the tongue is your lingual tonsils. You also have pharyngeal tonsils at the roof of your mouth. These are found, you see, these are your lingual tonsils, and behind them is your pharyngeal tonsils, and then in the middle is your palatine tonsils. These are mucosa-associated lymphoid tissue known as the pear patches. So you must also recognize that the lymphatic system is important for metabolism. How could that be? How could the lymphatic system be? Lymphatic system is important for metabolism. The lymphatic system is important for metabolism. How? Because you have a structure known as lacteals. What are these lacteals? Lacteals or lacteals. What are these? These are lymphoid tissue in the small intestine. Excellent. This is lymphoid tissue in the small intestine. Lymphoid tissue in the small intestine that is responsible for absorbing what? Who can we possibly be trying to absorb in our small intestine? What can we be trying to absorb in the small intestine? Nutrients. So what kind of nutrient does the lacteal help us absorb? How many kinds of nutrients do we have? How many kinds of nutrients do we have? Proteins, carbohydrates, lipids, nucleic acids. Who do you think lacteals help us absorb? Not proteins. Not proteins. What do you think lacteals help us absorb? Lipids. lipids. So lacteals help us absorb lipids. Lacteals help us absorb lipids. Now we move into our immune system. When it comes to the immune system, we have to now divide our immune system based on function. We said that the immune system is a vague collection. Like when you say, when I say the FBI, the Central Intelligence Agency, do you know exactly where is that? Which building is that? You don't know because they are a collection of different agencies working together. So the same thing with the immune system. So in the immune system, you have either innate immunity. What's another word for innate? That is natural immunity. There is innate immunity, natural immunity. There is also adaptive there's also adaptive immunity. Innate immunity is generalized, while adaptive immunity is specific. So innate immunity is your skin. Your skin is a barrier. It protects you from the entry of the pathogens and the infectious organisms. So the skin barrier. So one thing that protects our country from invasion of other army is the mountain. You know the mountain and the lake and the ocean? That's a natural barrier. So your skin is a natural barrier. It is rich in dermacidin, dermacidins, dermacidins. It's rich in acid mantle. This acid mantle burn all of the infectious microorganisms. So that's the type. But does that mean this is everything? No, because the bacteria can still penetrate. So in addition to the skin barrier, what else do we have? We have the natural killer cells. We have the neutrophils. We have the basophils. We have the eosinophils. Why are the basophils called basophils? Because they stain basic. They stain with the basic stain. You have eosinophils, natural killer cells, basophils, and you have eosinophils, and you have the complement system. What is this complement system? This complement system is known as the donut system. How could that be the donut system? Well, the complement system is called the donut system because it makes a donut in the wall. It makes a donut in the wall. It makes a donut in the wall of your pathogens. So you have bacteria, you have parasite, you have fungi, bacteria, parasite, fungi, you have worm, you have these different types of cells. When they invade your body, you have the complement system You have the complement system, and the complement system makes an assembly of proteins that look like they will make a donut hole. When they make a donut hole in the cell, in the body of the bacteria, what's going to go inside this hole? Water. So when the water goes inside the hole, the organism is going to be full of water, then it's going to burst and lyse. So the complement system causes the lysis and the explode. It will make explode the this dangerous infectious organism. It's going to fill it with water and make it to burst. So there is a cooperation between the innate and the adaptive immunity. You see, you have antibodies. These are a form of your adaptive immunity. And this is your innate. You see, your innate number one. Who did we say your number one? The skin barrier is like the ocean, is like the mountain, protecting your body from the army invading you, killing you, the North Korea invading you. The Kim Jong-un, he's coming, but he cannot because the that ocean. So same thing. Why aren't the bacteria entering your body? Because your skin barrier. That's a natural general blocker. Also the hairs, the mucus. These are all surface defenses. Then you have internal defenses. We mentioned those. We mentioned the mast cells and the basophils. They make the inflammatory response. What is the inflammatory response? The inflammatory response is a state of alarming. So the inflammation is a state of alarm. This is known as the inflammatory response. This inflammatory response is a response that is based on a trigger. Who is the trigger? The trigger is a invasion of bacteria, parasite, virus, fungi, worm, all of those, they represent infectious microbial organisms. These are the trigger. So your body, all of these, they have proteins on their surface. That's like their hair. They have proteins, they have lipids, they have sugars. They are on the surface. So that they are going to be detected. They are going to be detected with receptors. Receptors, these guys, the lipids, the proteins, the sugars, are going to be detected by receptors on the surface of the WBCs, the white blood cells. These guys, the neutrophils, the basophils, the eosinophils, then they're going to be coming to the site of the inflammation. So the inflammation is going to be 5 things. Rubor, that is redness. Calor, that is heat. Tumor, that is swelling. And you have pain, that is known as dolor, that is pain. And lastly is functiolasa, and that is loss of function. So any tissue that is undergoing inflammation must have, these are known as the cardinal signs of inflammation. What are the cardinal signs of inflammation? The cardinal signs of inflammation are 5. Number 1, redness, in Latin known as rubor. Number 2, heat, in Latin known as calor. Number 3, swelling, in Latin known as tumor. Number 4 is pain, in Latin known as dolor. Number 5 is loss of function, known as functiolasa. Known as functiolasa. So now, this tissue that is inflamed, like fire, the fire, pain, hot, redness, what's going to happen is you have your blood vessel here, right? That's your blood vessel. Your blood vessel. The blood vessel is made up of, what's the name of the cells that make up the blood vessel? We said this in lab. I'm sorry? Excellent. Excellent. These are endothelial cells. So whenever, let's say this is the tissue, that's the tissue, that's the tissue, that's your tissue. The tissue, let's say, has cell number 1, cell number 2, cell number 3, cell number 4, cell number 5. And what's going to happen is you have this white blood cell known as a monocyte. So now let's say bacteria comes into cell number 3. This is bacteria. This is bacteria known as cell number 3. That is known as bacteria. This cell is attacking cell number 3. Well, if there is bacteria, we say what's sprinkle? What is going to sprinkle? Like the crime scene investigation, we said the lipid is going to fall off, the bacteria. There's bacteria, there's virus, and you damage the cell. There is a war going on here. The same way when you hear a problem, you hear screaming, shouting, fire, sound, you smell something bad, the same thing in your body. When there's an infection to this cell, things are falling off. These things that are falling off, they make their way into the blood. Your monocyte, what do they have? What do the monocytes have?
Receptor.
They have receptors. So what are these receptors going to detect? They're going to detect invaders. They're going to detect the proteins that are falling off the invaders. These are known as cytokines. Cytokines. They're going to bind to these receptors, and then the monocyte is going to move and is going to localize right here now, about to enter. Why didn't the monocyte move to number 5, 4, 2, or 1? How did it just get to 3? That is known as chemotaxis. Chemotaxis. Chemotaxis, we talked about that. When you smell something delicious, you know how to walk to the kitchen. You don't walk to the living room. You don't walk to the bathroom. So you use your sense of smell to guide you to the site. So here, what did they use? They used their receptors to crawl. So chemotaxin is crawling to the site of the inflammation. When they arrive here, how are they going to go out? These monocytes that are coming to kill the bacteria, how are they going to get out? This is the blood. This is the intravascular fluid. Remember? So we are in the intravascular fluid. And now we're trying to make it into the tissue to destroy that. How are we going to go out? You see these endothelial cells? They're going to retract. They're going to retract like that. They're going to retract. When they retract, they open a gap here. This gap is known as endothelial gap or interendothelial cleft, because it's between the two endothelia. Then who's going to go out through this interendothelial gap? Who's going to go out into this interendothelial cleft? The monocyte. The monocyte is now going to differentiate into a macrophage. Remember, monocytes have to have to you see, monocytes differentiate into macrophage. Monocytes differentiate into macrophage. So then when they get to the site of the inflammation, they're going to become macrophage, and the macrophage are going to gallop up everything and kill the bacteria, remove the dead cell, remove everything, clear the path. And what's now going to happen? Healing. So now the new cell is going to come by regeneration. You have healing, and regeneration by entering into the cell division cycle to make up. So inflammation, because you have the interendothelial gap, is going to be a lot of fluid leaking. That's why you have swelling, tumor. Why you have redness? Because all the red blood cell, all the white blood is coming to the site of the inflammation. That's why you have redness. Why you have heat? Because you have a lot of blood rushing to this site. If you go to the lake, you put a thermometer in the lake. Then you go to the river where water's flowing. What is more hot? The thermometer is going to be at a high temperature where? In the lake or in the river? In the river. So here, the site where there's inflammation, a lot of blood is running to bring white blood cell. So it's hot over there. So that's why there's heat. There's redness because blood is rushing there. There's heat. There's swelling because there's edema. There's edema and water that is leaking out. Remember, we said that there's water inside the cell, intracellular fluid, water outside the cell, extracellular fluid, and water that is bathing the tissue. So this is known as edema. Then there is dolor, pain. Why is there pain? Because there is irritation of the nerve endings. Irritation of the nerve endings by prostaglandin. So prostaglandin prostaglandin is secreted, and it causes nerve irritation. So in inflammation, you have prostaglandins released and they cause nerve irritation, and the nerve irritation causes dolor, also known as pain. So that's why when you take NSAID, what do the NSAIDs do? The NSAIDs, non-steroidal and anti-inflammatory drug, they inhibit cyclooxygenase. Cyclooxygenase is going to inhibit the synthesis of prostaglandins. And when you no longer have prostaglandins, you will no longer have what? Pain. So when you take this aspirin, it is a non-steroidal anti-inflammatory drug because it blocks the synthesis of prostaglandins and it stops the dolor, the pain. And then there is functional loss of function. Obviously, inflamed tissue is a loss of function, the tissue that cannot do the function. So inflammation is the war that happened to remove all of the bad, the necrotic tissue. Okay, the mast cells, they do the inflammatory response. Number two, natural killer cells, they also do. These are all general, nothing here specific. The complement system does what? We talked about that. What does the complement system do? Please remember, we talked about that. What did I say? Excellent, excellent. Very good. Excellent. Everybody give a round of applause for Brittany. Excellent. 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 The donut hole, the donut system. So it assemble and put water in and then burst. The bacteria would blow up and the virus would blow up. Excellent. Good job. And we said that the phagocytes, these are the macrophage. What does phagocyte mean? Phago means what? Phago means eater. Site is that's macrophage. I'm sorry, macrophage. Phage is eater. Macro is big. These are big eaters, macrophages. But phagocytes, site is a cell. Phagocyte is a cell that swallows other cells. Monocytes, neutrophils, and macrophages. Excellent job, Brittany. Today, you're thinking very good. I'm going to give you extra credit. Excellent. Excellent. Good job. I am very proud. Very good. Okay. The innate immune system enhances adaptive immune responses so they can be more effective. Okay, so these are our general. Remember, all of this here, your skin, your hair, that's a surface defense. Then you have internal defenses. These are the mast cells and the basophils. These are the natural killer cells, the complement system, and the phagocytes. Then after that, you move into adaptive immunity. Adaptive immunity. Adaptive immunity is when you have T lymphocytes, antigen presenting cells, and the B lymphocytes. T lymphocytes, antigen presenting cells, and the B lymphocytes. These are, you see the T lymphocytes, they can either have T memory cells or T effector cell. The T effector cells, what's the difference between a T cell and a B cell? A T cell is like a soldier. It will go, it has a rifle, it will shoot the enemy. You shoot the bacteria, kill the bacteria. But a B cell is like a helicopter or an airplane. It will send a missile. It will not fight itself, but it will send the missile. And the missile that the B cell send is known as the antibody. It will send the antibody. Antibody. It will send antibodies. The T effector cells, that's why you have diseases like lupus and diseases like in the cartilage of in rheumatoid arthritis. That's the T cell causing granulomatous disease. You have antigen presenting cells, and these are B lymphocytes. So the B lymphocytes, they give us beef. You see, that's their weapon that I told you about. You see, that's their antibody. Does the T cell send anything out? They fight themselves. But the B cell, they send antibodies. So they both work together to defend you, but they work at different stages. First, you have to go through innate immunity. Usually, innate immunity is very effective. It destroys everything. But then adaptive immunity comes after that. And adaptive immunity will take over. That's the complement system. You see the complement system making the donut. You see the donut? See the donut of the complement system. So it has two pathways. There's the classical pathway that is used during the adaptive immune response. Occurs when the C1 react with antibodies that have been bound by an antigen. There is a classical pathway and there's also an alternative pathway. So for example, look at the classical pathway. Step number one. So this is the antigen. What is the antigen? The antigen is what the receptor caught. The antigen is what you are detecting. We said the police is looking for fingerprint. The police is looking for hair. The police is looking for DNA. So these are things that they could be traced back to you. But when the bacteria, the virus, the fungi enter your body, what we detect is known as the antigen. So now let's say we found that, there we go. That's you see the invading pathogen is all in green. That's your bacteria. That's your virus. That's your parasite. That's your fungi. On the surface, you see, just like that's its hair. That's the hair of the bacteria. What is that known as? Antigen. You see, the antigen is always some part that you can that you can trace back to your invading pathogen. Who's gonna recognize? Who's gonna recognize this antigen? It is the antibodies. And you have a protein known as the C1. So the C1 protein binds to an antigen. You see, that's the antigen, blue. The antigen antibody complex on an invading pathogen causing complement component, you have C2 and C4 to split in two. So this whole thing, all green plus orange, that's your C1. So your C1 is good. So first of all, you have your antigen, then the antibody bind the antigen. So now you have a complex. You have the antigen antibody complex connected together by what? The antigen. You have the antibody invading pathogen connected together by an antigen. That's the whole complex of three: the bacteria, the antigen, and the antibody. That's why they call it the complex. Antigen antibody complex. Right here, blue, yellow, green. Antigen antibody complex. Blue, yellow, green. Blue is the antibody, yellow is the antigen, green is the pathogen. Then who's gonna come recognize and bind this blue, yellow, green complex, this antibody antigen complex? Who's going to bind them? The C1. Then when C1 binds to the antibody antigen complex, what's going to happen to C2 and C4? They're going to break apart. When C3 and C4 they are broken apart, look, fragments from C2 and C4 that broke apart because C1 bound to the antigen antibody complex, they gonna combine to form a complex known as C3 convertase. C3 convertase. So C3 convertase here was made when C3 bound to when C4 bound to C2 after C1 bound the antibody antigen complex. Now let's see at what C3 convertase would do. C3 convertase is going to split into two right here. C3 convertase, after it bind together between C2 and C4, is going to break apart. And one of the fragments from the C3 is going to join C3 convertase to form C5 convertase. That's the C5. So you see C3A, you see C3 has broken into C3A and C3B. C3A is a cytokine. That's a cytokine. Is gonna attract. That's what cytokines do. They attract. They attract phagocytes to the infection sites, and C5b is gonna bind. You're gonna have C5 convertase. So C3b is gonna bind to the surface of the pathogen, and C5a and C5b, they're gonna make the donut on the surface of my green invading microbe, the donut. So a fragment from C5 is gonna join C6, C7, and C9 to form the membrane attack complex, which makes a hole in the pathogen's plasma membrane. So the water is going to rush into the hyperosmotic cytoplasm, causing the pathogen to lie. You see, water is going to move. Why does the water move inside to the pathogen because if inside the pathogen is very hyper solute. It's a high, so the water moves from an area of a high concentration to an area of a low concentration to burst. So the complement is to make this donut assembly. We talked about that. I just drew that. It's over there. You see it? It is not what I drew. Yeah, right here. That's exactly what's gonna happen. You see, you have your blood vessel. You have your blood vessel. Inside the blood vessel, what is flowing inside the blood vessel? The red blood cell. And who's that? That's your monocyte. Look at the bacteria attached. This green is the pathogen. You see the green pathogen attack here. Now what's going to happen? You have these blue cells. These are, the resident sentinel macrophages and mast cells. They secrete cytokine. So the cytokine is going to allow the phagocyte to come out to destroy all of the passage and remove the necrotic debris to begin the healing. They didn't show you the interendothelial gaps. So you see the inflammatory response is an event that results in warmth, heat. What did we call heat? Taylor. Redness, because a lot of blood rushing there. What did we call redness? Rubor. What's going to cause the pain? Dolor. Who's going to cause? Why do we have pain? The prostaglandins. Excellent. What do the prostaglandins do? How?
They're like a messenger that activates, binds with something and activates the receptor.
The prostaglandins make the pain. That's why we take the NSAID to block the synthesis of prostaglandin. But how do they make the pain? They do something. No? Hmm? What do they do? I think you're gonna get a lot of extra credit. What do they do? Yes, you said it. Yes, what do they do to the nerve cell? I said that. You know she's going like this. The nerve. That's what I said there. I said they irritate the nerve endings. Very good. Very good. They irritate. If you said irritate, you went like that. That's very close. But you said I said they irritate the nerve endings. Right here. Nerve irritation. Prostaglandins lead to nerve nerve irritation. Irritation. They cause a nerve irritation. That's what causes the dolor. That's what causes the pain. Pay attention. You're gonna miss out, guys, on the extra credit. So the mast cells, they detect injury to near. So this cell is injured. There's pathogens. Mast cells detect injury to nearby cells and release histamine, initiating the inflammatory response. So that's why they take antihistamines, because who's releasing histamine? Mast cells. The histamine increases blood flow to the wound sites, bringing in phagocytes and other immune cells that neutralize pathogens. The blood influx causes the wound to swell, redden, and become warm and painful. So the blood influx causes the wound to swell, redden, and become warm and painful. Okay. Now, this is the alpha beta T cell receptor. So this is a transmembrane region, and there are constant and variable regions of each chain. They are anchored by the transmembrane region. That is the transmembrane region. There is a constant region, there is a variable region. That is what's gonna help us detect. That's what's gonna help the T cell. This alpha beta T cell receptor is gonna help it recognize the through the alpha and the beta chains parts of the receptor to detect. So this is a antigenic determinant. You see this in the middle, that's the protein that we're gonna use to detect our antigens. So a typical protein antigen has multiple antigenic determinants shown by the ability of the T cell, thank you, with three different specificities to bind to different parts of the same antigen. So that's the same antigen of the bacteria. T cell number one bound to this site. T cell number two bound here. T cell number three bound here. So now we move into the antigen processing and presentation. So the first step is a pathogen or extracellular antigen is phagocytized by an antigen presenting cell, like the dendritic cell, and placed into a vesicle. Ingested pathogens are digested by lysosomes to extract their antigen. You see, we swallowed them inside. The pathogen has been endocytosed. Remember the endocytosis to the inside. Then a phagolysosome. You see the lysosome released during the digestion, and the antigens bind with major histocompatibility proteins that enter into the vesicle. These are major histocompatibility proteins to which the antigens are gonna bind. Then, number three, the major histocompatibility protein, now carrying antigens, are released from the vesicle and travel to the outer surface of the cell membrane. Then, number four, the dendritic cell is now presenting antigens which will activate the T cell that bind with the major histocompatibility protein. So does that make sense here? What happened here? An antigen right here, bacteria. So let's say there's bacteria out here that your body want to detect. The dendritic cell is going to take a fragment. It's going to take a fragment of it. It's going to take the fragment, throw it inside the core of the dendritic cell. When it's inside, there is lysosome. You know, the lysosome has the acid, the reactive oxidative species, proteolytic enzyme, degradative enzyme, lysosomal enzyme, knives, cutting, chopping up. So they're gonna chop up fragments of this pathogen. Then they're gonna take a tag, you know the tag, MHC2 protein tag, and tag this stuff we took inside. Then we endocytose one direction. Then we're gonna take it out and exocytose it out to present it. Look, we just got a fragment of a bacteria, we tagged it, and we're gonna put it to the surface. So now they're going to be put up here. So now the T cell, the T cell is gonna come and say, oh, this dendritic cell has arrested a pathogen and put on MHC2 complex the cuff, the handcuffs, and presented it to the top so the T cell can come take it to the precinct, destroy it. So these dendritic cells, we saw them already here. We saw them here, right here, dendritic cells. We saw them already here. Dendritic cell. You see, they look like spider because they're crawling, looking for bacteria. To take it, they have a very strange way of taking. They take bacteria in, swallow it, destroy it, take a fragment, add MHC2 tag to it, then present it to the top. So who can come and take it? T cell, T cell, T cell. Very good. You gotta go on home to be a millionaire. You see, T cell. You see? You see, T cell, T cell. The dendritic cell is gonna swallow the pathogen, is gonna destroy it, take fragment, add to it the you see the MHC2 protein tag. It's going to add it to it, then it's going to exocytose it here. So who can come and destroy it? The T cell. Good job. So now this is the differentiation of the T cells within the thymus. We're looking at the T cell destruction within the thymus. So that's how the T cell develops. You have untrained thymocytes from the bone marrow that enter the thymic cortex. There's a thymic epithelial cell presenting the MHC proteins um, to the untrained thymocytes, and there are thymocytes that successfully bind with the MHC proteins. So the immature T cells, these are called the thymocytes. These are little junior cells. They enter the thymus and go through a series of developmental changes that ensure both function and tolerance before they cleave and become functional components of the adaptive immune response. Thymocytes successfully bind with the MHC protein continues while those that cannot undergo apoptosis. So this is positive selection, selecting those that bind. Thymocytes that survive the positive selection are double positive, and these surviving thymocytes are all going to differentiate and to give us the mature T cells. There's clonal selection. Clonal selection is when stem cells differentiate into T cells with specific receptors called the clones. So the clones with the receptors, these are specific for antigens on the pathogen that are selected for and expanded. So you have activated T cells that are cloned and they destroy the infected cells in the body. And there are memory T cells that are produced and are remembering this encounter, and they would turn into activated T cells that will destroy future invasion. So these are dendritic cells. You see the spider crawling cells? They're presenting their MHC complex. Then the activated cell, T cell would bind to it, and then they would give us a differentiated form. What's the difference between a naive T cell and an activated T cell? The naive T cell becomes activated when it binds this MHC class 1 complex. So we present the antigens through the antigen presentation complex, and we have antibodies known as immunoglobulins. The same way the T cells have a light chain and a heavy chain, so do the B cells. We have different classes of antibodies. There are the IgMs, there are the IgGs, there are the IgAs, IgE, and IgDs. IgMs, their function, so the main antibody are the main antibodies of the primary responses, and they are best at fixing the complement. IgGs, these are the main blood antibodies of the secondary responses. And there are IgAs, these are secreted into mucus, and there are IgEs, these are antibodies of the allergy and the antiparasitic activity. And if a patient has a lot of IgE in their body, in their blood, you know that these are antibodies of the allergy. And that means they have a parasitic infection because that is the special type of the antibody that is present. So the same way we had clonal selection of T cells, we have clonal selection of B cells. So during a primary B cell immune response, both antibody secreting plasma cells and memory B cells both are produced. So these memory cells lead to the differentiation of memory plasma cells and the memory B cells during the secondary response. You see, that's the secondary response, and the memory cells, you see, not the plasma cells, the memory cells in the secondary response when we are attacked again with that same pathogen, it's those memory cells who will undergo this process. So when we look at the process of vaccination, when we give weak attenuated pathogens. When we give a very harmless, we give only the surface cytokines. So in an antigen, you just sensitizing your immune system to the surface antigens of that particular of that particular um of that infectious organism. That's how people work. They say criminal is always gonna look like this. They have this particular feat. That's how people make the stereotype. The stereotype say, the criminal, be careful. This person, you look like that. You look dangerous. So the same thing. You take the vaccination by sensitizing your immune system to certain proteins and sugars on surfaces that are not on your body. So the next time when you get the infection, so look, the primary immune response before the immunization was weak. But after you get a second exposure, you have a very rapid antibody secretion and an excellent immune response. So to elicit a response to a T cell dependent antigen, the B and the T cells must come close together. So to become fully activated, the B cell must receive two signals from the native antigen and the T cell cytokines. Okay. So, with the HIV, that is a virus that is able to do what? We talked about this in the beginning of the course. The HIV, the human immunodeficiency virus, has the capability of doing what? How does it affect your body? Yeah, so how does it replicate? Remember the central dogma of biology? We go from uh, DNA into RNA into protein. What kind of enzyme does your HIV virus have? Why do they call it the retroviral? They call it the retro.
Doesn't it work backwards?
Yeah, it works backwards. It's called reverse transcriptase. So we usually get mRNA from DNA, but this virus get DNA from RNA. Yeah. So there is a seroconversion, the rise of the anti-HIV antibody levels and the concomitant decline in measurable virus levels happens during the first several months of HIV disease. So in the beginning, a person infected with HIV, they would have a rapid, immediate response and then a slow down phase. So unfortunately, this antibody response is ineffective at controlling the disease. So the disease then progresses and rises again towards AIDS, in which all the adaptive immune responses are compromised. Why? Because who's the HIV virus target? Your white blood cell, your T cells, your B cells. So it's like imagine there's a disease where all the police die. So now crime is going to rise, and in the society there's going to be disorientation. But that virus, it does not target your bone cells, doesn't target your epithelial cells, doesn't target your muscle cells, doesn't target your nervous cells, it targets your white blood cell. So this shows the IgA immunity, and these are found in the nasal mucosa. That's the lymphoid tissue, and these are the pear patches of the small intestine that generate IgA immunity. Both use M cells to transport the antigen inside the body so that the immune responses can be mounted. These are your MHC2 complexes that help the T cell. So the MHC2 complex, only your T cell is blind. The T cell cannot see except the MHC2. There could be a bacteria here. If there's no MHC, your T cell will never recognize it. That's why the dendritic cell. You see, who's the cell that helped the T cell? It is the dendritic cell. Hypersensitivity is the opposite of immunocompromise. So sometimes you have some people who have, we said we don't want hyper, we don't want hypo. We don't want hypersensitivity. Now, pollen, is pollen harmful? Pollen is useless. But your body would assume that, oh, this may be virus, this may be bacteria. It will elicit a very intense reaction, hypersensitivity. The opposite is hyposensitivity, like HIV. You become immunocompromised. So hypersensitivity is mediated by there's IgE mediated, IgG mediated, there's the immune complex mediated hypersensitivity, and there's the cell mediated hypersensitivity. So we talked about these diseases, rheumatoid arthritis and lupus. They represent how you can have systemic symptoms where your T cells are damaging the cartilage of your bone and they are damaging your system. And we talked about erythroblastosis fetalis, and that is when your mom is Rh positive or negative. The mom is Rh negative, and the baby is Rh positive. So, does it happen on the first pregnancy or the second pregnancy? It happens on the second pregnancy. So the mom in the first pregnancy would have memory cells. So the second time, when she's pregnant again with another Rh positive baby, her antibodies, her immune system is going to destroy the baby's red blood cells. So during the birth, the Rh positive fetal erythrocytes leak into the maternal blood after the breakage of the embryonic chorion, which normally isolates the fetal and the maternal blood. The maternal B cells are activated by the Rh antigen and they produce large amounts of anti-Rh antibodies. The Rh antibody titer in the mother's blood is elevated, and the Rh antibodies are small enough to cross the embryonic chorion and attack the fetal erythrocytes. So erythroblastosis fetalis is the hemolytic disease of the newborn. Jamie once said that, right? The hemolytic disease of the newborn is the result of an immune response in an Rh negative mother who has multiple children with an Rh positive father. So during the first birth, you said the first birth, the fetal blood enters the mother's circulatory system and the anti-Rh antibodies are made. Then during the gestation of the second child, these antibodies cross the placenta and attack the blood of the fetus. So the treatment for this disease is to give the mother anti-Rh antibodies, known as a RhoGAM. So these are anti-Rh antibodies during the first pregnancy to destroy the Rh positive fetal red blood cells from entering her system. We talked about that. This is Kaposi sarcoma. It is a lesion that causes your immune system to damage all of the tissue that has been hypersensitive. Any questions?